This video will introduce you to a groundbreaking methodology in neuroscience called optogenetics. We'll explore its potential applications for studying and treating the neurological disorder called epilepsy. This video will focus on recent advances and future applications of optogenetics in treating epilepsy, and so it assumes that you have a basic understanding already. If you're looking for an introduction to the concept, you can check out the video linked in the description below. With that being said, let's go through some of the basics. First, what is epilepsy? Epilepsy is a neurological disease that is characterized by frequent seizures, or sudden spasms of excessive electrical activity in the brain. The intensity and impact can vary from one seizure to another, but they often manifest in uncontrollable movements, sensory disturbances, and even a loss of consciousness. Normally, cells in the brain, called neurons, fire electrical impulses in a controlled and coordinated manner. However, during a seizure, some regions of the brain experience uncontrolled bursts of electrical activity, which can spread across neural networks, causing a positive feedback loop where cells continuously signal and the normal mechanisms to prevent signaling fail. When this happens regularly, the condition is called epilepsy, and it can be caused by many things, like genetics or damage to the brain. The key issue in epilepsy is the disruption of the balance between excitatory and inhibitory neurons. Excitatory neurons activate the brain, while inhibitory neurons act like a brake, prevent excessive firing. In epilepsy, excitatory neurons become overactive and the brain's braking systems malfunction, leading to uncontrolled electrical activity. Optogenetics allows scientists to directly modulate the activity of neurons involved in seizure generation. By inserting light-sensitive proteins, called opsins, into the neurons, researchers can selectively activate or inhibit these cells in real time using light. This is done by delivering a gene encoding the opsin into specific neurons through a harmless virus, like an adeno-associated virus, or AAV. This can be done by having all the cells in a certain area express the opsin gene, or by targeting specific types of cells to express the gene. There are two main types of opsins used in optogenetic epilepsy research. There are channel rhodopsins, which are opsins that are non-selective ion channels, meaning that a wide variety of ions such as sodium, potassium, and chloride can pass through, that are activated by blue light and cause neurons to become excited. While primarily used to stimulate neurons in other research areas, in epilepsy studies they can be used to activate inhibitory neurons, enhancing their ability to prevent seizures. There's also halorhodopsin. These types of opsins are a type of ion channel that respond to yellow or green light to open and pump chloride ions into the cell. This causes neurons to become inhibited. By targeting overactive excitatory neurons, halorhodopsins can directly suppress the excessive firing associated with seizures. For epilepsy, researchers typically target inhibitory neurons to restore balance in hyperactive brain circuits. By shining light on these opsin-expressing neurons, they can enhance the activity of inhibitory neurons, essentially turning down the electrical overactivity that triggers seizures. Imagine a scenario where an individual is about to experience a seizure. In preclinical trials with animal models, researchers are able to recognize the onset of seizure activity by monitoring the brain's electrical signals. As soon as they detect abnormal brain activity, light is shined on the targeted brain region through fiber optic cables implanted in the animal's brain. When the light reaches neurons equipped with halorhodopsins, it inhibits the firing of these overactive neurons, essentially quieting the area of the brain responsible for initiating the seizure. This light-induced inhibition is instantaneous, halting the spread of abnormal electrical activity and preventing the seizure from progressing. On the other hand, when channel rhodopsins are used to activate inhibitory neurons, these neurons can dampen the excessive excitatory signals, restoring the balance between excitatory and inhibitory activity in the brain. A 2019 study investigated the use of optogenetics to prevent and halt seizures after they had started. In this study, researchers introduced halorhodopsins into neurons that connect the thalamus, which is a main relay station in the brain, usually associated with combining input from the different senses, to the cerebral cortex, the surface of the brain that has many high-level functions, like memory, language, and personality. The seizures the authors were looking at were caused by stroke damage to the cerebral cortex. Those are called cortical strokes. What the researchers found was that they could prevent seizures in real time. The researchers used real-time monitoring to detect the onset of seizures and then delivered yellow light to inhibit overactive neurons connecting from the thalamus to the cerebral cortex. Those are called thalamocortical cells. They found that by using optogenetics to inhibit these thalamocortical cells, they were able to halt seizures in their tracks after they had started and restore normal functioning. The authors also note that they did not aim to discover the exact site of the seizure initiation, but also, importantly, that they did not need to. In a quote from the authors, whether the seizure initiates in the cortex or the thalamus, we found that the thalamic output was required to maintain the cortical electrographic in behavioral seizure, which can be summarized to mean that all seizure activity that happened as a result of a cortical stroke required increasing activity in the thalamus to maintain the seizure. By inhibiting the seizure activity at the thalamus, the authors didn't need to target the specific area damaged by the stroke. This means that there is therapeutic potential for this type of treatment moving forward, as treatment might not have to happen at the area of injury. By disrupting activity in key areas, it could lead to general treatments for seizures. While much of the research so far has been conducted in animal models, optogenetics holds great promise for future human treatments. Current epilepsy treatments, like anti-epileptic drugs or surgical resection of the brain tissue responsible for seizures, can have significant side effects, and they're not always effective. In contrast, optogenetics offers a targeted, real-time approach that could minimize the adverse effects associated with broad-spectrum medications or irreversible surgeries. However, there are still challenges to overcome before optogenetics can be used in humans. One of those is delivery methods. One of the key hurdles is finding safe and efficient ways to deliver the light-sensing channels into the human brain. While viral transfection works well in animal models, ensuring safety in human patients is critical before clinical use. Researchers are currently exploring various viral vectors and non-viral methods to achieve this goal. Because epilepsy has many different causes, it's important to know where to insert these channels and into what types of cells for maximum efficacy. One of the second challenges is long-term viability. The long-term viability of the optogenetic treatment is another limitation. Since epilepsy is usually a chronic disease, any optogenetic solution to the epilepsy problem would need to endure during a significant amount of time without decreasing the quality of life for the patient. The third hurdle is combining optogenetics with brain implants. One exciting combination is the use of optogenetics with existing brain implant technologies. Devices like the responsive neurostimulation, for example, are already used today to detect and attempt to prevent seizures in certain patients. One day, such devices might employ optogenetic control, exerting their influence with much greater precision and much more effectiveness. Optogenetics has emerged as a promising leap in epilepsy treatment. Allowing the power of light to take the lead, scientists can, for the first time, control the real-time activity of certain neurons that cause seizures by finely tuning specific brain circuits involved in seizure activity. As research evolves, optogenetics may actually mark a turn in epilepsy treatment, offering even better seizure control for patients with fewer side effects than available with conventional treatments. The next steps involve translating these findings from animal models to human clinical trials, but the potential is clear. Optogenetics could revolutionize